We're gonna go ahead and get started with this last section here, and there may be parts of this that is more applicable to you than not. You're not going to hurt my feelings if you need to get up and walk around a little bit, but you were going to miss out on all the fun. So this last part is where we hand it off to you guys, and I kind of tried to prep you guys with an email to say, some of you guys have come and you're like, you've got some experimental ideas. You need to have a conversation about your project, number of samples, experimental design, something like that. We like to try to do that all together, and so that people can benefit from the experience and kind of see how this process goes. And then we'll do this for as long as need be. We're going to have a time tomorrow afternoon as well. So if you don't get in today, then you can jump in tomorrow. But that's the goal, is to let Rick and Stephanie and Mike share their wisdom, and then some of you guys, others may have a little bit more proteomic experience and be able to share some wisdom as well. But we need a brave volunteer. Who wants to start? Should we just start over here and just go around, however you guys want to do? I'm sharing the wrong screen, but all we're going to see is this. But yeah, that's great. Wonderful.
What exactly do you want us to do?
Who asked that question? All right, David, I want you to tell me about your experiment. Like, if you had some questions about experimental design or something that you're thinking about doing and how best to do it, then this, kind of like you had called about a week ago, and we had something like that with you personally, but.
All right. Well, I'm never hesitant to talk, unfortunately, for you. We've had great luck with these guys, and they've been very helpful at every step. We've been doing proximity labeling. Are other people doing that? It's been working amazingly well for us. I feel like proximity labeling is the new proteomics, at least for things that aren't strongly interacting with your protein of interest. Especially helpful if you work in a system like C. elegans, which is what I do, which you have to work pretty hard to bust them open, right? So preserving complexes is really difficult, and we study kinases. So we want to know things like substrates, and you're not going to get those in pull downs for the most part. One of the things I've been talking about a little bit is some confounding variability that we've gotten in some of our experiments, and I don't know what to attribute it to, because I'm not the hands sort of making the preps, although I expect it comes down to the preps. But that's not so much a question. I guess another direction we're moving into, and maybe you could talk, maybe this is best for tomorrow, but has to do with phosphoproteomics. Again, we study kinases, and maybe even combining, well, at least possibly combining proximity labeling and phosphoproteomics isn't entirely crazy either. Wondered what you might think about that.
I'm looking at you, Rick.
Yeah, it looks like everybody's looking at me. I don't think we've done any phospho enrichment on any proximity labeling, and so it should. I mean, basically, proximity labeling, you're expressing a local concentration of a ligase, and so you're biotinylating proteins around your protein of interest and then pulling them down. And so the only drawback that I see is that oftentimes we're starting with a small amount of material, and so if you're looking at phosphorylation, again, that's another small amount of material. And so typically the input for phospho enrichment is pretty high, because typically about 1% of the proteome are phosphopeptides. And so if we'll do an enrichment, a standard phospho experiment. Here I am sealing my own thunder for tomorrow. But we start with, a couple hundred micrograms of protein and digest that to peptides, and then enrich the phosphopeptides, and then we're basically injecting all of that. And so we're starving for material. And so my guess is we could probably see some phosphopeptides enriching from a pull down, but I don't know. I don't know how useful it would be. It might be more useful to actually look for the presence of phosphopeptides in those interactions. And that's something we don't typically do, but it's something it's certainly possible to do. We typically don't turn on those modifications for those type of searches. But especially if you're talking about a kinase, I mean, that makes sense, right?
Yeah.
To be honest, I have never thought of it. So I think that's probably where I would start, is not try to enrich that, but just look to see if we have phosphopeptides present there. And I'll plug Mike. I mean, that a targeted method, this is where, if you know that your kinase is active on a certain substrate, having standards of that peptide and then actually trying to quantify on it in a targeted method, that might make the most sense.
Yeah, so that would be great because we have some candidates and I can even guess at one site. And so if we've already done the experiment and the data is kind of sitting there, that could be great.
Can I kind of just ask your experience with the proximity labeling? Because sort of a challenge that I have in dealing with folks is a lot of times they'll read your paper and they'll do it once and they'll bring, how long did it take y'all to get it up and running to the point where you felt like you generated proteomics worthy samples?
Yeah, let me get up my PowerPoint. No, just kidding. It was almost immediately. So what we did is we knocked in turbo with CRISPR, because it's really, so one recommendation is always try to go for physiological levels if you can. And then, here was the really surprising thing. We've done it on several proteins, and some of them, when you do this experiment, you look to see if there's increased biotinylation over background. Usually there's some background because that's a thing that cells do. And in some cases, we didn't see much, and we almost thought it wasn't worth sending you samples. And we sent you the samples, and we got the most beautiful volcano plots you can imagine with like our proteins, the ones we knew we would get right at the top. So it didn't take, the most work was honestly just doing the knock-ins. It just does its own thing. And different systems may have different setups. We were just looking at steady state. We're not looking at a time course. So there's other ways of approaching it to do that. I'm trying to think of, we used the data independent and multiplexing, and, when it works, it really works.
Well, thank you for sharing that and for that unendorsed plug for the services here. He doesn't get any extra anything for that, but I appreciate that. But basically, the idea is, if you're confused, is if you want to share a little bit about what your kind of like what your work is, a little bit what you're looking for, and then kind of like then how do you translate that into a proteomic experiment? That's really the biggest challenge, I think, is because you guys know your biology, you know what your stuff is, but then how do you make that into something that we can that would be a good proteomic experiment? Yeah, please, Pat.
Can I follow up the question about phospho-MS? Because we study mitochondria, is quite hard to do the phospho-MS from the mitochondria. And another challenge is we are specifically look at tyrosine kinase. Even if we are looking at major tyrosine kinase, when I talk to the expert, now I'm not expert for MS, but when I talk to others, everybody tell me it's very difficult. And then when we send the grant to the study sessions, you have to do it. So back and forth. So how we can prepare so that we can detect them and also can detect the substrate.
Yeah, it's a challenge when for a phospho because we usually pick up more serines than we do tyrosines. And so, yeah, now you're looking at an even smaller proportion of
Exactly.
the sample material. So again, if you have some known targets, maybe trying to target something of interest for your study section, but I don't know if anybody, our big phospho guy left the room.
Well, so I'll stand in his place. This is what I know about the phosphorylation. So, tyrosine, it's less than 1%.
Right.
And so that's one of the things you can look at if you do a phospho thing, you can look at the number of threonines and serines and tyrosines. And so, yeah, I mean, it falls out pretty well.
You do a
So-
motif analysis and it's just a big S in the middle of your plot.
Yeah.
Yeah.
So you're talking about the challenging of the challenging. So, phosphorylation's 1%. And if you're looking at a very specific organelle, now you're talking about a subset of that. And then you're talking about a subset of the subset. So,
We want buckets of your material.
yeah. So I mean, that's a hard way to make a living, but I don't. We'll give him a chance when he comes back. But are you work with Art Solomon out there? Because he does a lot of tyrosine
And I thought
phosphorylation.
Art had developed some specific
Yeah.
creations
Yeah, I think he
Yeah.
to do the pull downs.
Started talking about tyrosine phosphorylation the second you walked out of the room, and
Yeah.
we have no answers here. Is there a way to specifically enrich for those that Art was doing or something?
There is. I mean, the conventional way is the cell signaling antibody cocktail. I forget what the number is, but it's a couple of antibodies for phospho-tyrosine. It takes, a huge input. It's pretty expensive experiment to do, but it works reasonably well. I mean, you need, milligrams of protein because, I mentioned phosphorylation is about 1% of the proteome, and tyrosine phosphorylation is about 2 to 3% of the phosphoproteome. And so you just don't see any tyrosine phosphorylation unless you enrich it specifically. But there are some SH2 superbinder domains that also recognize these phosphorylation sites. And so Art Solomon at Brown he didn't originate the work, but he's grown up a clone that you can you can enrich these, and literally they do the experiment for pennies on the dollar after growing it up. And so I can put anybody interested, I can put in contact with Art. I know the person who did it originally was not completely forthcoming with Art's group on the intricacies of growing it up. And so Art didn't appreciate that. And so after he got it worked out, he freely gives it to anybody that wants to. And my inability to do cell culture has prevented us from doing this here. So we need, a person to take on that role. But it's something that we eventually probably will do for enrichment because it's cost effective. And so I'm looking at Karen right now. So
Yeah, that's what I was thinking.
sorry, who asked the question? I apologize.
Hang
Yeah.
over here.
Okay.
I
Oh,
want to
yeah.
know-
So you know Art. Yeah. Okay.
I want to know more details because we have to do it.
Yeah.
The
yeah.
material is not the issue.
But yeah, I mean, I know of it. And so my small role in it is that we had a manuscript with Art where we use provanidate as a treatment of the cells. Provanidate is a phospho-tyrosine inhibitor. And so it basically inhibits the dephosphorylation. And so if you treat with provanidate, you see phosphotyrosine levels go up. And we use this in a TMT experiment in one of the channels to boost the overall signal. And so that signal may account for 90% of the phosphotyrosine signal of the TMT mix, but it allows that peptide to show itself above the noise level so that the mass spectrometer can switch on it, and then you can identify the things present at lower levels. So there are, some issues with spiking in a high level, but it basically opens up the analysis to be able to look at lower abundant species. And so I forget the title, but basically if you look at phosphotyrosine and boost,
Yeah.
that paper comes up.
Dennis, there's also a hand raised in there.
Yeah, I wanted to say, those of you that are on Zoom, this is your chance to jump in here as well. And it looks like Jill may want to be our guinea pig. I mean, our first Zoom person to talk about her experiment.
Sure. Just as way of introduction, I study yeast, so just Saccharomyces cerevisiae, and you guys helped me out with the previous project using the DIA approach to compare just basically changes in protein levels in different yeast strains. And now I want to work on the second batch. And so I'm kind of now that I know that 5 is the new 3, I'm kind of trying to figure out how to set up my sample set. And I realize that I'll have a more thorough discussion with you later, but I guess I'm trying to think about if I have 4 different yeast strains, for example, and I want to try each strain under 2 conditions. And when Stephanie was talking earlier, I was just trying to think about how to actually plan prepping those samples, given that I'm kind of limited to processing only 10 each day, or well, 2 sets of 10 each day. And so I guess if I could just hear more, is it better to, if I have an A and a B that are more closely paired, is it best to do like 5A and 5B on one day, and then a second 5A and 5B on the next day, and then kind of repeat the same thing with C and D, or is it better to kind of mix things up?
I'm killing time here as I'm watching Stephanie wrestle a battery in her microphone because she's the one that's going to answer this question, I'm sure. That's my stalling.
So let me get this straight. So Jill, you can do 10 at a time, and you can do 1 in the morning and 1 in the afternoon. So for those 40 samples, if we did
well, they all have to grow overnight, and then they have to grow for a while, and then one set will be at one temperature and one set will be at a different temperature. So the limiting factor is I'm limited to 10 at each temperature per day, essentially.
Okay.
And so I'm just trying to figure out, is it better to kind of just clearly define I'm comparing A and B and do 5 of those pairs each one day, or I guess, because I didn't realize before that all the DIA samples are run independently. So I'm just trying to figure out, is it better to do all of A in one day, or all of B in one day, or how
I
should
would do
think?
the mix of the A and the B. So you could do 5 of each on one
Okay.
day, 5 of the other on the other day.
Okay,
Again, it's kind of, I don't know if it'll matter a whole lot, but we do want to take note of that. So
okay,
whichever 5 you do on a day, say this is day 1, that's day 2.
okay.
And then we can check that also in your analysis. But
Okay.
yeah, if you can randomize that and have like both groups in
Okay.
that batch, that would be good.
Okay, yeah, because I'm just trying to figure out, I'm trying to compare the changes in B and D, right? I'm trying to change, I think
Yeah.
that, because I hadn't thought about that, and like I said, I'm sure I'll talk more with you later when we're talking about the details, but
Right.
increasing the sample size from 3 to 5 kind of throws off kind of what I had been thinking about, and so it just needs an adjustment. I mean, it's fine, and if that's what you think is important, that's value, and I'm glad that I heard you say that, and so I'm just trying to figure out how to actually implement it.
I had the same problem. So I'm not just analysis. I actually was wet lab. So I do understand the limitations of like physical preparing of samples. So just wanted to throw that out there. I am aware of the challenges.
Okay.
So yeah.
That's great. Thank you.
Thanks, Jill.
I have kind of a similar question, maybe going the other way. So I have 2 experimental setups, both kind of totaling out to 60 samples, because I have 3 sort of treatment groups in one of the experiments, 2 in the other, but then they're all across multiple cell lines. So I'm wondering, how much flexibility is there on an N of 5?
Cell lines is also something we haven't discussed because they're going to have different proteomes. And so
Yeah.
typically with like the library and the database search, we typically run those separately. But if we do with all of the cell lines together, I'm thinking through the Spectronaut software and how it's building that, because it'll borrow for the information across all of the cell lines, right? And
Yeah,
build
it's
that
not as
library.
much of a problem now as it was.
With the other-
Yeah, yep.
Yeah. We recently kind of switched our database searching parameters, so that's why I'm having to actually think about this. And I have a recent design on this where it's 3 cell types, 4 treatments, plus a gender effect. So that kind of becomes this multifactorial type process. And so way that one's going to work is if you have a control in each of the cell lines, we can kind of compare, say, this control from this cell line, this control from that one, and break it out, subsetting that way, where you're looking at the differences within this specific treatment across your cell lines. So that's one biological question. The other question would be, are there differences in the treatment within each independent cell line? And so you can put them all in the model, but you're not going to have enough replicates to really build the model with all of those factors. And it's probably going to break. Could just tell. So it's almost more of you do these independent different analyses, and now you've got sets of protein differences within each, and you can make your Venn diagrams of overlapping of what's consistent in this data set, what's consistent in that. It really is going to be what's our main question biologically. So it's throwing a hard one out there.
Yeah.
But
So do you think then, because I'm also then doing multiple time points, multiple cell lines,
time points
multiple
is another
treatments?
thing. So especially like time points, a lot of times right now we will do pairwise comparisons by time points. But if you have 5 time points or more, there's actually other models to generate for time data, like time course studies and things.
Right.
We haven't done that a whole lot. That's not typically a type of design that we've run,
Okay,
but I happen to have a project that I'm working on that has that right now.
gotcha.
So
So do you think with multiple time points, multiple cell lines, and multiple treatments that more than 5
yeah.
is necessary? How many
Yeah.
would you say is probably necessary?
Well, I don't even, I think the other one's gonna have 8 replicates each.
Okay,
That'd be a minimum.
gotcha.
Yeah, as
Okay.
many as you could get in there, I'd probably try to increase a little bit. And it also depends on like your effect size. So I didn't talk about that either, but how different you think your proteins are gonna be based on this treatment. If
Gotcha.
they're really small differences, you're not expecting a lot of change, you need more replicates.
Mhm,
If it's a really massive protein change,
mhm.
you can get by with a, smaller, the minimum. But with that many factors, you still want to have 8,
Mhm.
something like that. Yeah.
Thank you.
Cell culture is easier because you just grow up more stuff. When it starts to get into like tissues and patient samples, that's when we have more issues. Yeah. Good discussion. Put me on the spot.
You're doing great, Stephanie.
I always
More
feel like
questions?
we're gonna get in trouble because,
I was going to say, more questions for Stephanie. I mean, anybody else have something they want to share?
yeah, Joe.
Yeah, so just for background for everybody here, so my lab, we use rat models of vascular dementias and non-vascular dementia, all Alzheimer's related disorders. And so we have, a whole slew of different rat models. They're all in the same background though. So, and this is a completely non-hypothetical hypothetical, right? So in one of the sample sets that we sent already, we have 3 different transgenic models and then 1 wild type group.
Yeah.
They're all from the same time, but is it better to analyze them all together, or should we do individual comparisons of each transgenic against that wild type? Because right now in the analysis file that I have, they're all together, and I'm not
Yeah.
sure what.
Yeah, I think we do it all together because it's a matter of what pairwise comparisons we want to make. So if I'm going, say, transgenic 1 versus 2, I want to have all of the samples normalized together so that I can do all the different pairwise comparisons. So a lot of times we will just normalize it all together and then do all of the different comparisons. Sometimes it's been important if they don't want to go across, so you don't care about transgenic 1 versus transgenic 2, that's not a comparison we want to make. I would actually separate them out and just do the wild type versus transgenic 1, wild type versus 2 as an independent analysis. Because then you're removing that other group from our normalization, and it's going to change those normalized values ever so slightly. But you're removing that variability of the other factor and making a more straightforward comparison.
Sure. So
Yeah.
that's more of the questions that we want to ask, because essentially
Yeah.
what we're trying to do is biomarker discovery, right? So we're just trying to discover all the candidates that we can now, but really
Yeah.
what the question we care about is, okay, in this vascular dementia model, what's different from the wild type versus
Right.
I don't care if this protein is higher in this transgenic model than in this one. I care
Right.
only if it is higher compared to the wild type.
Yeah.
And so for that, maybe it's better.
For that one, it's better to separate them out so we can normalize them differently across
Okay.
each one. So, and we can do that. Yeah.
Great.
So, are you dead now too?
Yeah.
It's the end of the day. The batteries are dying.
Follow up on just, yeah, that's their way of cutting me off. I know you guys flipped a switch. So you said if your fold change is big versus your fold change is small, could you define that? Like how much, like your volcano plots cut things off
Well, it's not
intuitively
the
at
volcano
twofold. So
plot. It's actually the like overall number of proteins that are going to pass significance. So a lot of times if you have a really small effect size, not lots changing, if you get to that adjusted p-value, it may flatline because there's just not enough passing anything. And now if we try to adjust a small amount, the FDR is always trying to make it into a range of 0 to 1. And so if nothing is really coming out different, that's a small effect size. We need a lot more to kind of see those minor changes. But sometimes in treatments, like my triple negative breast cancer data, comparing triple negative breast cancer versus a normal cell line, everything is different. And so that's a really large effect. And you can see just hundreds and hundreds of protein changes in the proteome. But if I'm looking at a specific drug, you don't want everything to change because now you killed the patient. So it's kind of those, what your biostatistician looks at for effect size. Are we expecting just a large everything or kind of the smaller? Yeah. And a lot of that you won't know, and you're not going to know until you do some sort of pilot experiment. And that's why the pilots are really important. And so that's also kind of why we have our voucher system.
Yeah.
To give you some of that data.
In the real world, your expected fold change is calculated after you look at your budget and what the power.
Exactly.
You know,
And then you
the
write
power study says, like, oh, well, I know I expect a big change.
Yes. Because that's how you write your power calculations in your grant. Yeah, it's all wonderful.
Kind of combining those things. So I'm interested in the balance. So if you have a lot of parameters that you're looking at, but you also expect a pretty big effect size.
Yeah.
I work in triple negative breast cancer.
Yeah.
So I do expect if I am actually trying to induce like pro-metastatic signatures downstream of 2 different signaling pathways.
Yep.
So I would expect some pretty big changes. How would that affect, even though I have a bunch of parameters, like the amount of samples?
Parameters. So again, you could get by with like the 5-8 replicate more than likely.
Gotcha.
Yeah,
So 5 to 8 is probably okay.
that's probably a good range.
Okay.
But if you didn't have that big, large size, then I'm wanting more like 10, 15.
Yeah.
So I did a biomarker study. Actually, part of my PhD work was breast cancer, looking at bone metastasis biomarkers. And so what we did with that is had 15, gosh, going back, but 15 samples that had bone metastasis versus those that didn't. And you build a model and you're looking for any peaks, any peptides that come out that I just can't get rid of bioinformatically. So I threw every model at it. And this one little protein would not go away. And so that becomes your biological point of interest. And then what we did is took blinded patients. So you had another set where the same 2 different groups, use the model, classify them, and then they told me if I was right or not. And so that's another way to go about biomarker studies. with those methods, if you have a lot of clinical data, we can do a lot more stuff with it than just a LEMA t-test type comparison. So the way that data science is going now, everything's trying to go machine learning, all this type of stuff. That's really the power of that, if you can get enough replicates and something like that.
One thing I want to add, and I think we've talked around this. I don't know if we specifically mentioned it, but you can get by with an N of 3 on a cell line study on a small, a small comparison, but you can almost never get by with an N of 3 with an animal study. Even if you're asking one question, but just because of the variability with sampling or with the animal, with human samples, with cell lines, because there's so much, better controlled, so much less variability, you can get by with a smaller N, but the more complex the sample, or if there's any processing, any micro or macro dissection, then the N needs to go up just because the variability is going up.
Yeah, because I think our answers are kind of dependent on if it's cell culture, if it's serum, if it's tissue. Our favorite response is it depends. So yeah, all those things factor in.
I have a general question. You mentioned multi-omics before. So what specifically do you usually do other than proteomics?
So I also direct a bioinformatics core here that handles all the other genomics side of things. And then I'm an actual associate professor, and part of my research is multi-omics integration methods. So one of my last studies I did on triple negative was histone PTMs, phosphorylation, protein, DNA methylation, and RNA-seq.
Oh, wow.
So trying to put all that together and thinking about it with those different layers of biological information. Because with your RNA-seq, your genes and your proteins are not 100% correlated with each other. It's more like 50% maybe, and depending on what time you're actually capturing that information. Because genes are expressed in minutes, proteins are there for a couple of days. They're just not going to correlate that well. So a lot of the methods are based on correlation, which when we know they're not correlated is probably not the best methods we should be using. And so I've got a student right now who's working on some machine learning algorithms to actually run multiple different random forest, different models, and then rank those models and figure out the features that are important in each of the different layers. So yeah, it's an ongoing problem. And then my newest challenge is to throw in metabolites with that. And that's even more complicated because, like, just for me, where proteins can interact with genes, but that's kind of a whole different little beast that's hanging out over here. So you have to really go to the functional level and what pathways do we know? And now you're kind of having to interrogate biological function on top of the output of the expressions. So there's a lot.
That's what I was going to ask. I mainly do metabolomics and fluxomics and whole animal studies. I'm really curious, so proteomics is new to me, but I'd really love to get into it. And I'm used to doing all my own statistics, so this is kind of different for me. But are there any software you would recommend that we investigate if we want to do something that would join proteomics with transcriptomics?
Yeah, right now, like metabolite analyst is like, I think the best on the metabolite side, but it's kind of being done more manually with the pathway type stuff, as far as I know right now. There is a software that I've bought for the Embray called Big Omics Analytics, and they're working on the multi-omic platform of that. Right now, that will do all your RNA, your proteins. You can throw in your accounts file, and it does all differential expression. It does gene ontologies, pathways, biomarkers are in there. You can compare different data sets. Really powerful tool, but they're developing also the multi-omics part of that package. So yeah, I've just got that for our Arkansas Embray network. So I'm excited to start sharing that one.
Very cool. Thanks.
But, yeah.
So how much was that software?
It's depending on your use. So I'm paying more money for it. I met with the CEO and kind of set it up to where Right now I have 4 professional license, so I can host 25 projects in each license. And then that way I can control the analysis and then share it with my end user. But at some labs, I think are only spending a couple $100. It just depends on how much access, how many projects you're going to want to use. And it's all cloud-based. So you just access it through the web.
The name is what?
Big Omics Analytics. And I have a YouTube on that too. Yeah, we just recorded that last Friday with Big Omics.
Carrie, did you have a question?
Yeah,
It died.
yeah, I don't think with you. So I might be moving on topic,
That's okay.
but I did some proteomics 2 years ago, and we got results, and not one of them makes sense with my system, which is great. I've got a whole new world to explore, but I don't want to. So I'm interested in mitochondrial structure and FIFA. So my next logical plan was to using lysate, isolate the mitochondria, and then so that's what we're working now. But my question is, back several decades ago, when we would do pull downs and stuff like this, we would cross-link. Is that still something that people do for samples? Does it interfere with the mass spec? I'm just trying to figure out my next step if this was just fine.
For pull downs. So, I mean, there's a certain amount of cross-linking that can be done. Typically, most of it's overdone, but you know, if the cross-links are, you know, too heavy and can't be reversed with, you know, just acid, incubation, then it does cause problems.
Okay.
But it's a relative problem. You know, I'll say it caused more problems a decade ago than it causes now, just
Okay.
because the mass spectrometers are more sensitive now.
Okay.
And so we don't have to have all the material to analyze it. And so like the formalin fixed, you know, paraffin embedded tissue, you know, FFP slides that have been, you know, sitting around for years, there are cross-links there that we can't break. But there's enough protein that we can get that we can still make, you know, make
So if I'm
identifications.
just
Yeah, and you know, we've
not
got,
heavy-handed and find versions that are reversible,
yeah,
I should be, okay.
Yeah.
All right.
So I have a couple of software related questions. So I know you're now using Spectronaut, and Spectronaut has the ability to do normalization itself. So I'm just wondering, including global or local, you know, local within a sample or global across the experiment. And I'm just wondering if you've compared, you know, doing that normalization in Spectronaut versus your normalization after the Spectronaut output and what differences you see. And then I have another question about the multi-omics integration, but
Sure.
maybe answer that.
Yeah, so normalization is kind of a big deal, and there's so many different methods now. So that's why I prefer not to have the software do it, so that I have full control over how I normalize it. And then I can write the methods and we know exactly what happened. Because it's proprietary software, so there's certain algorithms in that can try to figure out, but can't. And so I'm still playing around with a lot of those parameters in the various softwares. I'm not the only one. And I don't know that we have a full conclusion on what we like with that software. So I tend to just turn all that off and get my raw values. That way I've got full control over the analysis and we can go from there. But the problem is some of the normalization methods require imputation, and I'm not a proponent of imputing missing values, especially with Lima. We don't have to. They're omitted from the analysis. So it's not requiring you to have a value, whereas a regular t-test does. So, and the package that we developed is actually comparing 8 different normalization methods. So we actually do kind of a quick evaluation of which one we're going to use for that data set too.
Yeah, thanks. And then, so my second question is about just, pathway analysis software, and if you've ever used Ingenuity pathway analysis. And so the other one was called Big Omics, I think you said?
Big Omics Analytics has way more than, so IPA is what we have here as an institution. The institution's bought into it, makes it really easy when you have access to it because you can see all the pathways and things. Big Omics actually includes that also in their software, plus way more. Like, I'll show it to you tomorrow. I'll pull one up and just kind of show you what all's in there. But they use Wiki Pathways, Reactome. There's a lot of other databases that aren't necessarily in IPA. But IPA has included metabolites. So that one's a really good one if we're trying to integrate between metabolites and proteins. But any of the pathway stuff is complicated when you get down to protein level because they're really built on the gene level. And so if a gene makes multiple proteins, we don't really know which protein this is in the pathway. And so it gets a little more complicated that way as well. You'll lose a bunch of IDs when you're trying to go to uniprot to gene IDs. Not everything matches. So that's one of the biggest bottlenecks with integrating data. You're going to lose a majority of your RNA-seq will not be there because we don't sequence as deeply. And then not everything has a matching uniform ID. So a lot of bioinformatic challenges are annotation. It's annotation and formats. That's probably where we spend most of our, and then you want to make sure you keep up with what version of the database you used. It's very important. It should go into the papers as too, because they're changing every month. The database updates monthly. And so I'm trying to keep up with the current version. So if you get a method write-up from me, you'll see there's a month and a year associated with that database. So I'm trying to mark which one we actually used. And if you're going to integrate between the genome reference and your protein reference, you need those to be in the same time and sync together. So annotation is huge, which I'm developing I have another student who developed a snake make workflow trying to do proteogenomics. Say you have whole genome data mutations at your gene that you're interested in, that's not going to be in your protein database. And so we can take that level of information, build a custom protein database that now includes your genetic variants within the database. But then the other problem is trypsin going to be the right enzyme? Because that amino acid change is probably going to be in a non-tryptic peptide, which I just had. And so part of our tool at the end is to run an in silico digestion with other enzymes. So you might need to use gluci or something else to pick out those variants, to have a shot at it anyway. So those are lots of things to think about.
Does the Big Omics software, so like one of the things that I like about IPA is it goes beyond just pathway enrichment and actually makes a prediction about activation or inhibition
the
based
regulation.
on, based on the actual directional change in your genes or your proteins. And so does Big Omics, because I know Reactome doesn't do that, or some of the other things like Bioconductor and R, it's all just enrichment versus actual predictions about.
Yeah, I don't remember if that one's in there. I mean, they have a whole biomarker linking to different databases and specific stuff, but I'm not sure that maybe, I'm not sure if that one's in there or not. Yeah.
Can I ask another question or just bring up another thing that might be of interest to people generally, which was that one of the things I hadn't anticipated is that, when let's say you have a control and a test, that it might be, the protein might not be detected at all in the control.
Yeah.
And in which case it's an infinite fold change. There's no p-value and it's not on your volcano plot. And we actually had quite a few things of interest, like maybe some of our best things were actually on that list. And I guess we ended up just deciding not to even try to integrate it on there because anything we gave would be false. But how much do you encounter that and what do you suggest?
Yeah, it's kind of when you're looking for lower abundant proteins are going to be that range. So you kind of have this range of really high abundant proteins we can detect really well. You're starting in transcription factors. All the things we really like to see biologically are kind of the lower abundant range. And now you start running into limits of detection. And so then it becomes almost more of a presence absence. And we can't say it's not there, it's just not detected. So you have to be careful. A zero doesn't necessarily mean it's zero, but it's just not detected by the instrument. So And then that would be something that, okay, we have biological information about, this looks like it's going to be really important in my system. And then we kick it over to Mike and see if we can't target it and validate that things are there. Yeah. Yep. Anybody else on Zoom have anything? I think all our mics are dying around here.
So if you're really interested in quantitative levels and you're detecting multiple proteins from a particular, or multiple peptides from a particular protein, presumably you feel safe in making the call with the most abundant peptide detected?
Go ahead, Rick.
Yeah, so the different software use different numbers of peptides for quantitation. One of the standards is using the 3 most abundant peptides for a given protein. And that's based on historic empirical data that shows that randomly the 3 most abundant peptides for any protein correlate pretty well with the abundance of that protein, just in distribution of amino acids. And so, but where there's sometimes difficulty is if those peptides are shared, if 2 of those peptides are shared with one isoform and one is unique to another isoform, then it's when we're rolling up the protein lists, we're using Occam's razor to make the smallest list of unique proteins that we can. So we're assuming that we see 2 peptides to this one protein and 1 to the unique isoform. We're assuming that all those peptides are to the same isoform, the abundant one there. That may not always be true, but if we don't have any other corroborating evidence for that second isoform to be present, we're not going to let a shared peptide make its, claim that it's there. And so that's one of the difference between, you'll see identified proteins and protein groups.
Right.
And the groups is the smaller concatenated list of the isoforms.
And
I
you'll
don't know if
see
that
those-
answers your question or not.
Yeah, and you'll see those IDs in the same list in that row. So you'll know which ones are grouping together.
And presumably, mass spec quantitation is now the gold standard against which everything else is measured. I mean, how was it established that, this method is the new standard?
I think we're the first to admit that the quantitation that we give is essentially a relative quantitation. So we're quantifying, that abundance of that protein in one sample relative to the next. A targeted method like what Mike's doing, he may spike in a protein and be, quantitative relative to that. We heard at lunch some of the aqua methods from Thermo, where you've got quantified heavy isotopes of a peptide, and then you can measure the signal of that peptide to the native peptide in your sample, and that comes closer to absolute quantitation. Although I would probably still argue that it's an approximation. It's not absolute, because it's only absolute if you assume that 100% of your protein was digested with trypsin, and 100% of those peptides were cleaned up with your, C18 cleanup, and 100% of those peptides got injected out of your auto sampler into the mass spec, and and so on and so on. So it's still relative. I love how they use the term absolute in the name. The aqua is absolute quantitation of, I forget the acronym, something, but yeah, it's a little presumptuous, but that's what we do as mass spectrometrists. We're a presumptive bunch.
Okay, but I want to get to this question up here, and then it looks like Ram has one back there. But Emery online said, just a quick question I meant to ask earlier. I work with fission yeast, and I was wondering if there were known components of yeast media that I should be wary of for sample preparation. For example, you should you said we should ensure there's no FBS in mammalian cell culture. Well, just to say, we just want to make sure that the FBS gets washed out. So I think you can't, it's hard to grow it without it, but yeah, how you yeast folks, what do we need to do with that?
Just the same thing. Yeah, just rinse your pellets. Yeah.
Okay, same sort of protocol.
Yeah.
PBS. Great. Thank you, Emery.
So this is more of a grantsmanship question. What kind of preliminary data we can show? If we are performing, let's say, quantitative proteomics. So if I'm saying that I'm having 3 samples, and if I'm doing a pilot, then the experiment itself is done, right? So when I write a grant, what is that one figure which I should show as a preliminary data?
The volcano plot. Yeah.
Yeah,
Right. So
so it's-
the volcano plot, when your protein of interest is far to the right and up. So, but you know, in cases where that's not, you know, the case where it doesn't look as good on a volcano plot as something else, a heat map looks, you know, does a good job.
But then like the reviewer will say that you have already done the analysis, right?
Not usually.
Yeah, no, I don't.
So let's say, let me put the question another way. So I'm struggling with amplifying the sample amount, you know, so for me, the challenge is to show that it is feasible. You're getting my point? Like, so let's say if you require 100 microgram, I'm not even able to reach like 10 microgram. So for me, I don't want to show like volcano plot, you know, I will have to show that like I'm reaching that amount, right?
It's a protein depth question, like how many proteins are you able to identify with a small amount of material? Kind of what you're trying to answer.
Yeah, maybe, or, you know, if you get a specific, and maybe we need to talk more specifics, but if you know that a certain protein is changing between your control and treated, but you want to see what's more, I mean, you know, show a Western, right? I mean, that's good for showing differences on one. You know, back to the quantitative issue, I still find great humor in the fact that, you know, Western blots are by definition not quantitative, yet somehow they became the quantitative gold standard in, you know, molecular biology for, you know, 30 years. So.
Is it a particular protein that you are looking for?
It's a family of protein.
So that family of protein expands within a certain range of size, like kilodaltons?
Yeah.
So then again, you know, you might have to concentrate it, the amplicon, and then like Rick said, you know, run it on a gel.
Okay.
And show that, you know, that range of size, you know, protein size is there in your, and that probably would be enough. And then you can say that I'll do mass spec with that, and I'm going to get this.
Yeah, you can do a
Set of proteins within that. So that way you're.
Okay, did we get that? Did we get you sort of taken care of there?
Yeah.
Okay, sorry, I was doing a battery change. Yes.
I just wanted to thank Sam and Stephanie. So we did get a pilot voucher. And one thing that I learned from the experiment that we did is to utilize the KISS philosophy. So we ought to make it simple, and that is what will get us to our goal. It was very, you know, Stephanie was very articulate. I know that the experiment that we did did not immediately give us what we wanted because we had done.
Stephanie, I had a question and a possible suggestion. I can't remember if it was you or Dennis that showed the output table for the peptide data with multiple columns and references back to the protein database. But do you have a YouTube video that shows you interacting with all of those data elements and what they mean and how to go about that.
I do. There's
Okay.
one that we did on, I think where me and Dennis are on that video together on the idea channel, going over Lima outputs.
So
Yeah.
could you send us or Dennis send us a link to that?
Yeah, we'll find that
Yeah.
specific
We're
one.
going through the differential analysis.
Because you're going to do it tomorrow, right? It might,
I'm gonna go
yeah,
through
she-
the whole
Yeah,
output.
She's going to
So
go.
it'd be helpful if we had a preview to,
Yeah, it's I think it's one of the top ones that's viewed on our idea channel. It just says Lima output. I think
It's also one of the oldest ones too. I don't know if there's a
it was one of the
correlation
first ones.
there.
Dennis cornered me and said, we're making this video,
Couldn't get
which I'm
anybody
glad
else.
he did.
Right.
You're gonna put it behind a paywall soon, right?
That's
Yeah,
right.
Subscription model, yeah.
What really makes me mad is when I go back and look at my own videos on YouTube and there's a commercial in there and I'm like, whoa, it's being monetized? Where's my cut? Where's my share?
No.
Okay, before, there is an online question, some just, it says, we said, okay, Samjana, do you want to go ahead and just ask your question or do you want do you want me to read it here?
Yes, I'm sorry. I mean, yeah, I wanted to go back to the study design question because we are doing aging studies and we have this genetically modified probiotic, and then we want to see its effect in the aging mice model and aging rat models, I'm sorry. And we have 3 different groups. One is the control, one is the SAM group, and one is the experimental group. And each group consists of 10 rats, 10 animals under the study. And there are like 5 males and 5 females. And then we are doing the study for a total of 3 months and collecting the samples once every month. So altogether, we'll be just collecting the sample 3 times from each individual animal. So will this kind of study design be sufficient for analyzing the proteins from the plasma that is related with the age signaling pathways?
I have a question. Is the age brought in, is it like a categorical variable where it's young, old, or is it actual numbers?
We are at first we are just taking the 10-month-old rats, and then we are just looking for a 3-month duration study. So there will be 10 months and then 11 months and then the 12-month rats.
Yeah. Okay. Yes, because I could turn that into like a factor, like a categorical variable to add age into our statistical model. So
Okay.
one of the questions would be like, how is this treatment effect dependent on age?
Yes.
If it's age dependent, that becomes an interaction type model. So you actually put both of those factors into this interaction, which is basically that question, is one dependent on the other? And then that will tell you which proteins are dependent on age.
Okay.
So
I might add
Yeah.
that if you're interested in signaling pathways, plasma and serum's probably not the best sample to be interrogating, right? So I mean, these are going to be low level. I mean, thought of at least as as low level that are probably going to avoid detection just because of dynamic range.
Yes. Okay. Yeah, we will figure that one out. I mean, like with our PI, we will have a yes. Anyway, thank you for the information.
Yeah.
I have a question. Why not just use the max quant for the DIA analysis? Why the spectrum now?
We started with max DIA.
Dennis, you want to take this one?
I could never get max DIA to work.
I couldn't either.
I did it to
And this is what I do for a living.
Why?
We couldn't get it to run.
Oh,
It just can't, yeah.
too much data.
The only papers outside of Jürgen Cox's group, the one that developed it, and they can get it to run, and every other paper with it has a, paragraph thanking Jürgen and his group and
For
helping
running
them to get it to run. And so it's not ready for dissemination.
One of the troubles with Max Quant 2 is I want to put it on a Linux environment so I can queue up things and make it run. And they say it's Linux compatible. It is not. I worked with a Google team to put all our stuff into Google Cloud, and the Google engineers couldn't get it to run either. So I didn't feel so bad.
You know, we say that and we love Max Quant for, you know, for
TMT,
IDs, for TMT. That's what we actually use that. And so I've been using the Max Quant software for years. Yeah, long time. But yeah, I was excited when the Max DIA came out, and just we can't get it to run.
It's kind of the same with scaffold DIA. I really like that approach, but it just crashed and still have a support ticket out for that. And so we moved on to Spectronaut. And Spectronaut is, you have to have a license for, but it has a graphical viewer to see all your fragment ions. Diane, you'll start seeing all, that's everywhere. That's the free version to run DIA database searches, but you have no graphical viewer, no confidence in seeing those fragments.
Yeah, but for a free software, Diane,
free.
incredible.
Yeah.
Yeah, it's great.
So
Easy to run. Even I can do it.
yeah, if you want to run, and that one includes phospho and that 2 other modifications. So, and that one will run on Linux.
I have 2 real quick questions. One is we have RNA-seq. We have a bunch of RNA-seq data from like 3 or 4 years ago for different cell lines. If we were interested in doing a prep for proteomics to do like a multi-omics integration, are there specific things for prep that I should consider if we were wanting to ultimately integrate this with existing
Just
RNA-seq
get your
data?
best proteomic data set you can get.
Okay.
Yeah.
So
So-
just same prep, but just better.
Yeah.
Cool.
Yeah. Because you're going to normalize those different, like you're going to take care of normalization in your RNA-seq experiment, and you're going to do protein normalizations. I do those separately because they're accounting for different technical factors, and they're just, and then once I have my normalized data sets, that's when I do the integration.
Okay.
There's a tool online, Mixomics,
Mhm.
that have all kinds of methods for integration of those data types. So
Gotcha.
I use a lot of their tools.
Okay. And
And
then
then MoGSA, the multi-omics gene set. It's not gene, it's singles, whatever. It's a gene set enrichment analysis tool for multi-omics. But that one, your samples, you couldn't do it like you're doing now. You actually have to prepare your samples from the exact same sample set to do that pathway analysis. So it's requiring, otherwise you introduce too much variability and stuff and the correlation breaks down. So if you want to do that, you have to prepare RNA-seq and proteins from the exact same samples.
gotcha.
But there's other tools now.
So Mixomics, I could
Yeah.
do that. Okay. And then another question for protein quantification. I saw that there should be that before you send your sample, and then you also do it here in-house. What is the ask for that? Just like a regular old BCA? I just take some of the pellet and then do a BCA. And what sort of like numbers are you all looking for?
For normal analysis, we ask for 100 micrograms, you know, knowing that, you know, there are some cases where that's not going to happen, right? I mean, if you're isolating a fraction, you know, a rare subregion of the brain on a small animal, okay, yeah, maybe you're not going to get that. But 100 micrograms lets us use our, you know, most reproducible techniques in the lab to digest it. And so the reason we would like for you to do the protein quant is we'd rather for you to realize that sample 4 is low than have us come back and tell you, hey, sample 4 is low and there were only 3 in N of 3 in that group. What do you want us to do? And so that, you know, it's pushing back on the researcher. And that's again, what's great about an N of 5 is that, oh, well, let's drop it. We still got 4 and let's go on. And it's always going to be the control, right? Always going to be the control. Yeah. So the question was, if we inject more sample, you know, do we can we see more things? And we can't up to a certain limit. And so the capacity on the chromatography that we use in front of the instruments is roughly a microgram and a half. And so if we inject more than that, it's the chromatography is going to degrade and we'll actually identify fewer peptides. And so that's kind of the maximum amount that we can inject. And so that's why, you know, sample enrichment's important. If you're interested in nuclear proteins, you know, do a nuclear prep, right? Now that's getting rid of all the other proteins of interest. And so it's going to introduce variability, so increase the N, but then you're looking deeper into the proteins that you're really interested in. Sure. So the question's about aberrant proteins or you know, mutated proteins, cross-linked proteins, fusion proteins, and so you can see them by mass spectrometry. You know, as a rule, I'll say we're not going to. And you may think, you know, I'm a pessimist, and I keep telling my wife that I'm not. I'm a realist, and there's a difference. But the reason I say that we're not going to is because on average, you know, across the 5,000, 6,000, 7,000, however many proteins we identify in a study, we see about 30% sequence coverage across there. And so if we see 30% sequence coverage, that means that 70% of the protein sequence we don't see. So I say there's probably a 2-to-1 shot that we're not going to see your peptide of interest if it's a fusion protein or not, right? And so now if it's a fusion protein and it's lower abundance, now that number keeps going down and down and down. It's possible, but it's not likely.
Anybody else? We talk ourselves out. I got a couple of announcements that we need to make before we close up for today.
Can I just say one thing?
Yeah, please do.
I was curious how many people are using, like, first, a lot of proteomics, we're looking for protein-protein interactions, at least for some, right? How many people are using AlphaFold to look for that and modeling interactions? So not many. Is there a reason, or is there just not?